So, dear participants, I welcome you all once again to the second session of this Atala sponsored FDP. So, today we'll be having a lecture on databases and molecular parameters, which I'll be giving to you. I think I don't need any introduction since I have been with you all since last 3 days. It's our 4th day of contact. So we are a research group working on the area of computer-aided drug design and other allied areas in bioinformatics. So today we'll give you an insight. I hope you can see my slides and hear my voice. I am audible enough. So we shall be discussing about databases, how to make databases pertaining to such computer-aided drug design, pharmacy, or life sciences fields. And Dr.
वहां से मांगो बोल रहे हैं उससे वापस जो सामान मंगाया था जिससे हल्दी चंदन सारा सामान भेजा था
Firoz Khan
2 लाख रुपए दिए थे
talked about QSAR. So
रशीद से 1 महीना पहले बात हुई कि बाकी का सामान भेज दे। सर ने बुला के बोला सर 2 दिन में भेजता हूँ, 2 दिन नहीं आए अभी तक।
We'll be discussing with the molecular parameters of QSAR modeling. So I would request all the participants to mute themselves. So now what we have is, what is a database? So database is a collection of related data elements which are held in the form of tables, which consists of columns and rows. What in the language of computer science, columns are said to be fields and rows are said to be records. Records are retrieved using a query language. Database technology is a well-established technology. We all have been using different databases in different fields for our research or daily course. So in the database, the basic entities is a table, which is basic element of information to track. That is, for example, if you are making a biological database, you want to put information about a gene, an organism, or a sequence, or you want to put citations. So in the columns, which will be attributes of tables, what we call here as a field, example for citation table, you need to have columns for title, the journal name, volume, and the authors. Rows are the records where actual data is fed, whereas fields describe what data is stored. The rows of a table are where the actual data is stored. So database is a very simple form of, like, if I give you an example, it's simply like a filing cabinet where you keep your records. So in the filing cabinet, as you can store many different records like sheets of paper, each containing multiple data elements. Similarly, in a database, you can store many informations regarding a particular topic. The biggest problem with the filing cabinet is that you can only store your data one way. Means it's one way you keep your data, but how others will see it, that is defined by you yourself. Here, there is no good way of searching your files based on any other criteria. But in a database, we are taking an example of a filing cabinet of invoices. The filing cabinet is a table. The columns are the fields of data on the individual invoices, which consist of customers information, product information, price, quantity. The rows or records are the individual invoices. So a flat file database is a spreadsheet. It is the electronic analog to the filing cabinet. This is more easily searchable than a paper file cabinet, but is still very unwieldy, especially for large amount of data. So there is a need to make databases for any such information, which should be handy to use and store huge data, and it should be user friendly. Users can use it very easily from their own desktops. So suppose you now want to be able to send an advertisement to every customer who bought the Atmos machine. You could add a column to your table that includes the address for each customer. But this is very inefficient. You will keep repeating information for customers who make multiple purchases. As the number of rows and columns grows, searching a flat file becomes more and more time consuming. Also, it is difficult to construct complex queries. For example, customers who buy the snow machine and who like opera or live in the southwest desert. So when your number of queries are increasing, it becomes difficult for a flat file to make such a database. So the solution is the relational database. A relational database contains multiple tables and defines the relationships between them. Thus, you might have now two tables, a customer table and a product table. So you join them through schema. So this is a database schema where you are making smaller multiple tables and they are being joined so that you can keep more information in such a database. So the databases these days are being made online, means they are made and then made available online so that people can search them. So when you query an online database, your query is translated into SQL, the database is interrogated, and the answer displayed on your web browser. So your computer and the browser are the clients, and there is a software to receive and translate your instructions to the database. So here we are considered about biological databases only. So online databases, there are over 1000 biological databases. They vary in size, quality, coverage, and level of interest. Many of the databases are of different origin, different kinds, and they are available online for the people to search, to make use of the information contained in them for their research and personal use. So they vary in size, means the amount of information they carry, quality, coverage, and level of interest. So many of the major ones are covered in the annual database issue of nucleic acids research. So if you want to find out databases pertaining to biological functions, you can go to nucleic acid research database issue and you can find the names of databases where the newer found databases would be added. So now question comes, what makes a good database? So the first quality is comprehensiveness, accuracy. It should be up to date. It should have good interface. We should be able to batch search things and download things. and it should have a connect to API. So the biological databases are of primary type, secondary type, and there are tertiary, quaternary databases as well. So primary biological databases are also called archival database. Since it archives, what happens is the experimental results submitted by the scientists are fed into it. So the primary database is populated with experimentally derived data like genome sequence, macromolecular structures, etc. So the data here entered remains uncurated. No modifications are performed over the data. So whatever the experimentalists find out through their wet lab experiments and acquire the data, that data is collected and stored in primary biological databases. Primary biological databases obtain unique data from the laboratories, and these data are made accessible to normal users without any change. The data are given accession numbers when they are entered into the database. The same data can later be retrieved using the accession number, and this accession number identifies each data uniquely and it never changes. So if you have to find, you have to make search again and again, you can use the same accession number. So examples of primary databases are nucleic acid databases such as GenBank and DDBJ. Protein databases such as PDB, which we have already seen that you need to download your protein PDB file for docking and all procedures. So PDB, which is a protein database, is a primary biological database. SwissProt, PIR, Tremble, and Metaseq. So now we move to secondary biological databases. So the data stored in these types of databases is the analyzed result of the primary databases. So what happens over here? The information contained in a primary database is taken and used to analyze some sort of result, and when the information in that experiment is again kept in a databases, such databases are known as secondary databases. Computational algorithms are applied to the primary database, and meaningful and informative data is stored inside the secondary database. The data here are highly curated. Processing the data before it is presented in the database. So a secondary database is always better and contains more valuable knowledge compared to the primary database. So we have seen that primary databases contain information from experimentalists and the data is non-curated. But in the case of secondary databases, the data is curated now, means it has been used to find out some other information, and that in many more information has been gathered. Now the data is being curated. So a secondary database is better and contains more valuable knowledge compared to the primary database. So examples of secondary databases are InterPro, where you have the knowledge about protein families, motifs, and domains. UniProt, which is a knowledge base of sequence and functional information on proteins. So then there are composite biological databases. The data entered in these types of databases are first compared and then filtered based on desired criteria. The initial data are taken from the primary database. So what happens is, if you have information on certain topic in different databases, and if that information or the initial data is taken from the primary database and then they are merged together based on certain conditions, means certain similarity metrics. So it helps in searching sequences rapidly. Composite databases contain non-redundant data. Examples of composite databases are OWL, NRD, Swissprot, and Tremble. So here we are showing you a figure in which you have lot of biological data. There is lot of genome sequences and there are sequencing centers that keep sequencing the genome. Then there are curators, and then you have a genome assembly, and then we have databases like GenBank, which are regularly updated by the help of submitters who keep submitting their newer findings and their new gene sequences. With the help of all the computational knowledge, use of different algorithms, different databases are being made. So now, what type of biological data you will use based on the type of information it carries? So they are broadly classified into genome databases, where the genomic information is held, sequence databases, where you can find the information about genome sequence, microarray databases. There are chemical databases where you have information about different chemical compounds which you can use in the area of drug design. You have all the information about that chemical compound in such a database, ranging from solubility to its pharmacokinetic or such parameters, where you can correlate the physicochemical parameters with any area of your pharmacy. Means you are designing that drug. Structure database, where you have information about the structure of a particular compound of a particular nature. Pathway databases, where you can find out different pathways that occur in a body, ranging from different organisms, from smaller organisms to Homo sapiens. Then we have disease databases, where you can have knowledge about a disease, its cycle, its drugs, its symptoms, its cure, the therapeutics that is involved in it, the diagnostic procedure that are involved in it. So you can get knowledge about every aspect of the disease from such databases. Then there are enzyme databases, where you have full information about a particular enzyme, its role, its inhibitors, its enhancers, promoters, every knowledge, its three-dimensional structure, how its subunits, almost every knowledge about it will be kept in such a database. Then there are literature databases, where you have all the information about literature, what has been done on it in the past. So entire literature review you can get from such database about a research topic. So we have now a look over few important bioinformatics databases, Gene Bank, which holds nucleotide sequences. The website is given over here. Ensemble, which has human genome, mouse genome, and many other genomes. You have PubMed, which has got literature references. NR, which has protein sequences. SwissProt, which is on XPASI channel, having protein sequences. Interpro, which is a database from UK. It holds protein sequences. Interpro, which holds protein domains. If you want to find out any domain of a particular protein, you can visit Interpro. OMIM, which contains all the information about genetic diseases. Enzymes has all the knowledge about enzymes. You can make use of it in your drug design if you want to inhibit a particular enzyme. PDB, which has all the structures of almost all the proteins. So you can download your protein molecule from PDB database for drug designing. KEGG, which has which is a database of metabolic pathways in different organisms. So if you are searching to find out which metabolic pathway is responsible for a particular disease, you can go to KEGG pathway and find out the metabolic pathway, the enzymes responsible, the enzymes taking part in the pathway, which you can find out what the malfunctioning of an enzyme is causing a disease, or what is undergoing in a particular pathway. Then till now we have studied about biological database. So to design a drug, you must have a target which you find out or you identify from biology, and then you must have a ligand or a drug molecule which you identify from your previous knowledge of chemistry. So to perform a drug design exercise to make such databases, or you are searching such databases for development of a drug, you need to have knowledge of both of them. So from biological databases, we now come to the chemical databases. So what is a chemical database? A chemical database is a database specifically designed to store chemical information. This information is about chemical and crystal structures, spectra, reactions and synthesis, and thermophysical data. So if you want to design a drug, you can download the chemical structure of a chemical compound from here, the crystal structure, how it looks like in 3 dimensions, the spectral characteristics identified by experimentalists, the reactions and synthesis procedure that it has undergone, how to synthesize it, or what to synthesize more from it. Both the knowledge is contained in the database, and its thermophysical data. Now, the types of chemical databases include chemical structures, the databases which have chemical structures in 2 dimensions or 3 dimensions, literature databases where all the literature related to the compounds is stored. You can get references and the work that has been done on a compound, you can find out from there. Now we have crystallographic databases where a compound's crystallographic data is stored, means the techniques that are being used to find out the crystal structure of a particular compound and its results, they have been performed by the experimentalists and the data has been provided to them, and this data is stored in crystallographic databases. So they are primary databases. NMR structure database. Nuclear magnetic resonance spectroscopy is a technique to find out the structure of a particular compound, and experimentalists perform it on every novel molecule that they synthesize or every molecule which is isolated from natural products undergoes NMR spectroscopy to find out its structure. So once you download a chemical structure from chemical structure databases, to identify that your structure that you are using is that structure only, you download information from NMR structure databases and compare it. So every molecule has its own spectral characteristics in NMR spectra. No two molecules have similar NMR spectra. Based on this fact, you can compare your chemical compounds. Then there are reaction databases. These are databases of all the reactions that a particular compound can undergo, or people around the globe have done such reactions around this molecule. So from this you come to know what kind of reaction your molecule will undergo or has undergone in the past, and what would be the novel reaction that you want to perform. Then we have thermophysical database, where all the information regarding its physical informations and thermal informations, what is the heat of formation, what is the heat of vaporization, what is its enthalpy, entropy, everything about it is given. So now we'll have a look at few major public chemical databases. So one is most often we use is PubChem. So it is a chemical information repository at the US National Institute of Health. So we have given you the address of PubChem. It's a public repository of information on small molecules and their biological activities, developed and maintained by the National Library of Medicine, an institute within US National Institute of Health. So when it was launched in 2004 as a component of NIH's Molecular Libraries Roadmap initiatives, it has been rapidly growing and now serves as a key chemical information resource for researchers in many biomedical science areas, including Bioinformatics, chemical biology, and medicinal chemistry. PubChem contains more than 235 million depositor provided substances, 94 million unique chemical structures, and 1 million biological assays, which cover about 10,000 protein target sequences. So PubChem provides various research and analysis tools for efficient use of this amount of data. So here we are showing you a glimpse of PubChem. So if you want to find out, supposedly you want to find out about aspirin, so here it will give you the whole details about here its chemical name, acetyl salicylic acid, its IUPAC name, 2-acetyl oxybenzoic acid, then molecular formula, molecular weight, IUPAC name. Then if you want to use this Smiley's format for different functions of computer-aided drug design, for example, either for docking purpose or for developing a QSAR model, you can copy these Smiley's sequence from here, these keys, then all the SDF formats and all you can Then you can go and download it. If you want, you can search it in entries as well. Then you go to substances, you can go to pathways in which it is involved. You can go to bioassays and you will find what bioassay to perform to estimate it, literature and patents related to it. So the PubChem database will give you an entire idea about that chemical compound which you want to find out against any other protein, or you want to find out about its activity information. So another is Chem Spider. This is a chemical database integrated with Royal Society of Chemistry's publishing process. So we have given you the address of Chem Spider. It is a free chemical structure database containing information on 34 million structures collected from approximately 500 data sources. It provides information on chemical reactions through Chem Spider synthetic pages. It uses a crowdsourcing approach that allows registered user for manual comment and correction also. It is owned by, as I told you earlier, Royal Society of Chemistry, which publishes approximately 40 peer-reviewed chemistry journals. It is integrated with the RSC publishing process, whereby newly synthesized chemicals or newly identified chemicals published in RSC articles, they become available on Chem Spider. So here is a glimpse of Chem Spider. Now here, if you want to search for aspirin, it gives you the details, same details, and then you can find out the derivatives also, aspirin related molecules, or what we call similar molecules. Then we have Chembl. Chembl is a chemical database which uses literature extracted biological activity information. Here, like PubChem was a chemical database where all the physico-chemical data was contained. Here what you get from Chembl is you choose a particular chemical compound and you will get its bioactivity data from here. It is a database developed and maintained by European Bioinformatics Institute, EBI, which is part of EMBL, European Molecular Biology Laboratory. So the core activity data in Chembl are manually extracted from the full text of peer-reviewed scientific publications in selected journals such as Journal of Medicinal Chemistry, Bioorganic Medicinal Chemistry Letters, and Journal of Natural Products. So whatever experimentalists report in these journals with their scientific findings, such novel molecules with a particular biological activity are contained in Chembl. From each publication, details of the compounds tested, the assays performed, and any target information for these assays are abstracted. Chembl also integrates screening results and bioactivity data from other public databases such as PubChem Bioassay and information on approved drugs from the US FDA Orange Book and the NLM's Daily Med. So Chembl looks like this. Now again we are searching for aspirin. It gives everything. Now it is giving you the information about it, the drug mechanisms, what is its action type, it is its target's name, target type, every binding site data. So every other data related to its biological activity is contained in Chem Spider. Now Another database, Kebble, a dictionary of a smaller molecule entity. It stands for chemical entities of biological interest. It is a freely available database of small molecular entities developed at the European Bioinformatics Institute. The molecular entities in KEBI are either natural or synthetic products used to intervene the processes of living organisms. As a rule, KEBI does not contain macromolecules directly encoded by genome. So it does not contain information like about nucleic acids, proteins, and peptides derived from protein by cleavage. KEBI provides standardized descriptions of molecular entities that enable other databases to annotate their entries in a consistent fashion. So KEBI focuses on high quality manual annotation, non-redundancy, and provision of a chemical ontology rather than full coverage of the vast chemical space. So here the KEBI has the data on chemical compounds that are of
ऐसे मूर्ख बच्चे हैं सर, हमने उसको मेल अभी मैसेज किया है, देखिए
that are of biological interest.
क्या आपको क्या बोल रही थी? आपको कुछ की है? आपको कुछ की है?
Please mute yourself.
अटेंशन सीकर है, मैं साफ शब्दों में बोलूं, अटेंशन सीकर है।
इनको पता ही नहीं है कि हमारा एग्जाम है क्या है।
हमने सर इनको अलग से टाइम टेबल जनरेट करके दिया है, आप देखिए जरा। और हम क्या करें, हम लिख दें इनका पेपर जाके, इनका पेपर अब हम ही लिख दें। वो क्या लिखी है मेल में, मेल में बकवास लिख के भेजिए, ये
फॉर्मेट में बोलिए उसको।
हमने अलग से ग्रुप में मैसेज भी तो डाला है। अब सो रही होंगी तो हम क्या कर लें सर?
So, sorry, there was an interruption. So, while Campbell focuses on bioactivity of a large number of bioactive molecules, KEBI is a dictionary that provides high-quality, standardized descriptions for a relatively small number of molecules, currently approximately 50,000. Now, this is how the KEBI looks like. If you want to search for aspirin, it gives you the biological information about it for which aspirin is responsible. Like here it is given aspirin triggered protectin D1, then here it gives you aspirin triggered resolvin D2. So whatever information you want, biological information about aspirin, it is showing you. Now comes the major public chemical databases. NIST web book. It contains thermodynamic and spectroscopic data of chemicals. It is compiled by the US National Institutes of Standard and Technology, and it compiles chemical and physical property data for chemical species and distributes them through the website called NIST Chemistry Webbook, whose web address we have given over here. These data include thermochemical data, example, enthalpy of formation, heat capacity and vapor pressure, reaction thermochemistry data, enthalpy of reaction and free energy of reaction, spectroscopic data like IR and UV visible spectral data, gas chromatographic data, and ion energetics data. So NIST webbook looks like this, and here you just see we are searching for water, so it gives its formula, IUPAC, gas registry number, chemical structures. Then you can come here and you can find gas phase thermochemistry data, condensed phase thermochemistry data, phase change data, gas phase in energetics data, mass spectrum, vibrational and electronic energy level, gas chromatography, fluid properties. So if all these data you want for a particular chemical compound, you can go to NIST webbook. So here we see if you are enthalpy of phase transition you want to see, you can see the values. Critical density of liquids and gases, if you want to see, we are searching in the case of, we have taken a simple molecule to be water. You want to find out about its triple point temperature, enthalpy of formation, boiling point, coefficient of expansion, Joule-Thomson coefficient, density. All these physicochemical parameters can be found about a molecule on NIST webbook. So then we come to Drug Bank. It is also a major public chemical database. It has comprehensive information on drug molecules. So currently we are involved with computer-aided drug design. We want to know which molecules have been enlisted as drugs, and for drug repurposing purpose also you need to download molecules and have a knowledge about these. Since we all know that they have undergone all the clinical trial procedures and they are safe molecules. So for to find by the use of computer-aided drug design, if you want to repurpose a drug, this is the best database to be used. So Drug Bank is a comprehensive online database containing biochemical and pharmacological information about approximately 8,000 drug molecules. It includes US FDA's approved small molecule drugs and biotech drugs, which are protein or peptide derived, as well as experimental drugs. Drug Bank provides a wide range of drug information, including drug targets, mechanism of action, adverse drug reactions, food-drug and drug-drug interactions, experimental and theoretical ADMET properties. ADMET means absorption, distribution, metabolism, excretion, and toxicity. So here it contains all the pharmacokinetic and pharmacodynamic parameters as well. Most of these data are curated from the primary literature sources by dominant scientific experts and skilled bio-curators. So Drug Banks looks like this. If you want to find out, like we have given aspirin again, so what is its toxicity level, what are the lethal doses, which pathway it inhibits, what are the enzymes concerned, all this information is contained in Drug Bank. You want to know the pharmacology of the molecule, you can find it over here. Interactions, what are the products available in the market, under which category it is sold, chemical identifiers, what clinical trials it has undergone. Every information of it is given in this database. Now, among the few major public chemical databases is HMDB, the Human Metabolome Database, which is a comprehensive information on human metabolites and human metabolism data. This database contains curated information derived from scientific literature as well as experimentally determined metabolite concentrations in human tissue or biofluid. Biofluid here means urine, blood or cerebrospinal fluid. Reference mass spectra and nuclear magnetic resonance spectra for metabolites are also provided when available. In addition to data for detected metabolites, which have been measured concentrations or whose experimental confirmation of existence is present. The HFDB also provides information on expected metabolites, those for which biochemical pathways are known or human intake exposure is frequent, but the compound has yet to be detected in the body. Then comes ToxNet, which has a collection of toxicological information. ToxNet is maintained by National Library of Medicine at National Institute of Hospitals, National Institute of Health. It's a group of databases covering toxicology, hazardous chemicals, toxic releases, environmental and occupational health, and risk assessment. So 16 databases have been integrated into the ToxNet system, and users can search all these databases either at once or individually. Three important databases of ToxNet are Chem ID+, which is a dictionary of over 400,000 chemical records; the Hazardous Substance Data Bank, which focuses on the toxicology of potentially hazardous chemicals, providing information on human exposure, industrial hygiene, emergency handling procedures, environmental fate, regulatory requirements, and all these data. The Comparative Toxicogenomics Database, which contains manually curated data describing interactions of chemicals with genes, proteins, and diseases. This database provides insight into the molecular mechanics of underlying variable susceptibility for environmentally influenced diseases. Now, the protein database, it's an archive of experimentally determined 3D structures of large biological molecules such as proteins and nucleic acids. These structures were determined primarily by using X-ray crystallography or NMR. So PDB is not a small molecular database. It contains the three-dimensional structure of many proteins, at some places with some small ligand bound to it. So the PDB are maintained by Worldwide PDB and freely accessible via the websites of its member organizations: PDBE in Europe, PDBJ in Japan, RCSB PDB, and so on. So a PDB data bank looks like here. You can search a PDB database for knowledge about a particular protein. Few other chemoinformatics specialized databases are Available Chemical Directory ACD, Zinc containing more than 460 million compounds, Maybridge containing more than 6 million compounds, Medcom, Beilstein, World Drug Index, World of Molecular Bioactivity, Toxic Substance Control Act. Now comes the second part of the lecture, the molecular parameters. Molecular parameters are essential part of a QSAR model, means which molecular parameters you will consider for making a QSAR model. So the molecule has to be considered as if you consider it as a one-dimensional entity, or you consider it as a two-dimensional entity. Nowadays we consider it's in the historical development, we consider it as 3D entity. So if you consider it as 1D entity, then you have information of atom counts, the molecular weight of it, the number of functional groups. You want to have this information. When it is considered to be 2D entity, when we draw it on a paper, it has topological indices, V-cut, topological polar surface area, Shannon entropy, and all these informations. When we consider it as 3D in our QSAR programs, it has geometrical parameters, molecular surfaces, parameters calculated in quantum chemistry program, and so on and so forth, many number of molecular parameters. We will take the case of here few molecular parameters that have to be considered in the area of QSAR. So, number of atoms, absolute and relative numbers of carbon, hydrogen, oxygen, sulfur, nitrogen atoms in the molecule, number of bonds and whether they are single, double, triple or aromatic bonds, number of benzene rings, number of benzene rings divided by the number of atoms, molecular weight and average atomic weight, number of rotatable bonds, hydrogen bond acceptor and hydrogen bond donors. These simple descriptors reflect only the molecular composition of compound we are not considering the geometry or electronic structure of the compounds. So these are simple parameters of 1D. So when we consider topological parameters, if you consider a molecule to be in 2 dimension, then these are the edges and these points are the nodes. So descriptors based on the molecular graphical representation are widely used in QSAR and QSPR studies because they help to differentiate the molecules according to size, degree of branching, flexibility and overall shape. So here you define what we have read in chemistry. What is a iso compound, what is a neo compound, whether the compound is cyclic or so on. Now there are geometrical parameters. Like you want to find out moment of inertia for it, then you do rigid rotator approximation and the moment of inertia characterizes the mass distribution in the molecule. You calculate shadow indices, you find out radius of gyration, area, it is a molecular surface area descriptor. This may describe van der Waals area of the molecule. It is related to binding, transport and solubility purposes. Now steric parameters. So if you put your molecule into a box, this will be its breadth, this would be a length. So finding out length to breadth ratio LB1. Then finding out molecular thickness, ovality, molecular volume, putting the molecule in a box and trying to find out its volume, stereomol parameters and Taft steric parameters which are needed to form a QSAR equation. So the oldest equation in QSAR is that of Hamid, where your benzoic acid is undergoing hydroxylation, forming the benzoate ions. To find out the lipophilicity of it or the sigma parameter, what we call Hamid's parameter sigma, you have to find it out mathematically, and to determine the solubility of and the polarizability things of the particular molecule, we need to find sigma, and it is one of the descriptors to be calculated for building a QSAR model. So sigma values for every substituent depends upon its position, whether it is placed ortho, meta or para in an aromatic moiety, and for hydrogen all the positions are same, so it has similar value, but for different other substituent it has varying values. So if there is a molecule whose sigma values you want to calculate, Then there are available tables on which you can calculate manually. Now comes Taft equation and what we call ES, the Taft parameter, which is quantified steric special effects using the hydrolysis of esters. So these steric effects were quantified by Taft parameter. It is a rate constant for ester hydrolysis, and Taft parameter ES is also considered in QSAR model development. So this ES parameter, it has different values. So H is usually as the reference substituent, but sometimes when another group is used as reference, the value would be considered 1.24, and then at the methyl position, if we replace it by propyl, tertiary butyl or chloro, the compound will change and its activity will change, and this with the development of a QSAR model or what we call a QSAR equation, you can predict the activity of different substituents. Then one is P, what we call octanol water partition coefficient, or sometimes denoted by pi, which is a parameter in the development of drugs in the wet lab as well as with the approach of bioinformatics. So it is a hydrophobicity parameter where we see the concentration of drug in octanol versus concentration of drug in water. It is a comparative value and it defines whether the molecule is hydrophobic or hydrophilic. The value of log P, if it is greater than 0, the compound is hydrophobic. The value of log P, if it is less than 0, the compound prefers polar media or is polar, or we call it hydrophilic. Now we do Hansch analysis, where biological activity is a function of these parameters. So biological activity is related to log of 1 by C. C is the concentration of the drug. And here in framing QSAR equations, we make use of sigma, the Hamid constant for every substituent, pi, the hydrophobic substituent constant, log P, which is octanol water partition coefficient, and Taft steric constant. So when we used Hansch analysis for framing QSAR equations, we consider electronic, hydrophobic as well as steric parameters. So this kind of 3D QSAR modeling or framing 3D QSAR equations, we consider all the three types of molecular parameters. Then while doing studies on chemical compounds, we observe their quantum parameters as well. Quantum chemical descriptors include some observables that can be computed by means of quantum mechanical approaches. So the most important are calculation of different types of energies, calculation of dipole moment, molecular polarizability, what is the molecular electron density, finding out highest occupied molecular or highest occupied molecular orbital and lowest unoccupied molecular orbital energies when you are predicting whether a chemical compound will undergo a certain kind of reaction. The binding of the drug to the receptor is also dependent upon electronic spread, development of energy across them. So to find out what would be the mode of binding of your drug to the receptor protein, you need to have the knowledge of all these energy parameters. And the atomic charges, as we know, like charges repel and unlike charges attract. So when there will be development of charges on your molecule, there is possibility of binding to the receptor. So you must have knowledge of these quantum chemical descriptors to design a QSAR model. Dipole. The dipole moment is a measure of the bond polarity in a molecule. The distribution of electron density is not uniform in case different elements combine, and we define dipole moment mu is equal to QR, where Q is the charge magnitude and R the separation distance, and the dipole moment is expressed in dBi. So here we have given you, like in the case of methane CH4, the net dipole becomes 0 as all the hydrogens are distanced equally and they have equal charge on them. Molecular polarizability. Polarizability is the relative tendency of a charge distribution like the electron cloud of an atom or molecule to be distorted from its normal shape by an external field. Application of an external field induces a dipole moment. So mu is equal to alpha E, where alpha is the polarizability. Polarizability values have been shown to be related to hydrophobicity and other biological activities. So when we develop a QSAR equation, we consider this parameter as well. Molecular electron density. The electron density is the measure of the probability of an electron being present at a specific location. Usually when you use different softwares, it shows you like this in the colored way, where the electron density is the most. So we know that to carry out a reaction to make a bond, electrons participate in it. So to know where these electrons are maximum or where the electron density is maximum, where it is minimum, where does the electron cloud lie, to do reactions to predict its properties, this knowledge is of utmost importance. So here what it is showing is the electrostatic potential for benzene moiety. The more red an area is, this one, it has higher the electron density. The more blue, it has lower electron density. So this electron density provides indication about the most reactive sites in a compound. It also provides useful means for the detailed characterization of donor-acceptor interactions. Those people who must have done docking studies, it shows binding with the receptor protein, and those bindings are either by hydrogen bond or donor-acceptor interactions or pi interactions or hydrogen bonds. So this descriptor has been employed in QSAR studies to describe drug receptor interaction sites. Then comes the Hobo Lumo energies. It is the energy difference between the energies of Hobo orbitals and the Lumo molecular orbitals. The transition between Homo and Lumo is observed in some chemical reactions. The smaller is the gap, the higher is the reactivity of the compound, means it's easier to fill that gap and move to highest occupied molecular orbitals. Homo-Lumo generally describe reactivity and can be responsible for high toxicity. Atomic charges. The partial atomic charges are due to asymmetric distribution of electron in chemical bond. Charge-based descriptors are widely employed as chemical reactivity indices or as a measure of weak intermolecular interactions. As we have shown it over here, that when it is existing in its different forms, now we can see the atomic charge at different atoms is different. So the calculation of partial charges relies on the Mulliken population analysis and is carried out also by means of semi-empirical quantum methods. Energy. So what is happening in a molecule? You have different atoms. Atoms are undergoing different motions ranging from vibrational, translational to rotational motions. And what is happening is we are having different kind of energies. So there is a stretching energy involved in stretching mode, bending energy, torsion energy, and non-bonded interactions energy. So if you see, the molecule is looking like this. So these two atoms, the bond is getting stretched and you have stretching energy to the magnitude of this. Bending energy, the molecule is bending where these three atoms are involved by an angle theta. You can calculate bending energy with this formula. Non-bonded energy, the two atoms that are non-bonded are also interacting by this radius ij. So you calculate van der Waals term and the electrostatic term, and then you call these two energies together to be non-bonded energy. Now, with the help of knowledge of these molecular parameters and knowledge of to make our database, we have reported in CBIO where we have made a knowledge base of antimicrobial peptides. So what we did in this was we have taken antimicrobial peptides from human microbiome. So what we did was we assembled metagenomes, downloading them from NIH Human Microbiome Project in FASTA format. All these acquired metagenomes were subjected to Prodigal, which is a fast and reliable protein coding gene prediction tool for prokaryotic genomes to isolate the protein coding regions of the metagenome. BioPython was used to isolate peptide sequences below 50 amino acids. And we made use of MPEP, a sequence-based prediction tool for antimicrobial peptides, and it was used to predict and isolate all those peptides that show potential antimicrobial activity. So the MATLAB implementation of the MPEP source code was used for the prediction. So we identified a number of antimicrobial peptides from each body location, ranging from airways to GI tract to oral buccal cavity, skin, and urogenital tract. 96.2% of the antimicrobial peptides where 36.2% of the antimicrobial peptides were left undefined or unidentified. The position of finding them could not be identified. So we used OpenBabel to generate the PDB file containing the coordinates of linear peptide strands from the amino acid sequences, and to study peptide folding, Bromax was used to find the stable energy conformation of these linear structures. Then when we found those stable energy linear structures, we did energy minimization until the maximum force is less than 10 kilojoule per mole, employing steepest descent algorithm. We conserved ensembles of NVT and NPT to equilibrate the system at constant volume, temperature and pressure of 1 bar pressure, 300 Kelvin temperature and for 100 picosecond each. Then we performed molecular dynamics simulation using the leapfrog algorithm with an integration time step of 2 femtoseconds at constant temperature and pressure of 300 Kelvin and 1 bar respectively for 1 nanosecond. Then MySQL Server version was used to implement HAMP. The web interface and other functionalities were implemented using HTML, PHP, Python, and JavaScript. So then an incorporation of an intelligent search engine using locality sensitive hashing was done. And in this, we have a screenshot of the search page where you can find 3D model and similar sequences of antimicrobial peptides from human microbiome. So now I end my presentation over here. So if there are any questions, so if you have any, do you have any questions to be asked? So we have ended with the talk. If there are any questions, you can ask me. Are there any questions that you would like to ask about molecular parameters and 2D or 3D databases? So I think there are no questions to be asked. Okay, so Dr. Santosh asks that free software is used or paid. So there are some freely available softwares that I have shown over here to you that you can use to make your own databases. And for generating molecular parameters, you have eDragon as a freely available software. So to the participants, once again, I would ask if you have any more questions, you can write it in your chat box. I would feel happy to answer them. So are there no questions to be asked? Then we can end this session and meet at 2:30 p.m. for post-lunch session, which would be a hands-on experience. We'll be giving you training on homology modeling and the use of Swiss param server, as one of the participants had asked yesterday. So I think we stop here in this session and we'll meet again at 2:30 p.m.